now let's discuss neurophysiology. If you remember from the chapter 3 when we talked about cells, or really chapter 1, we talked about characteristics of living things. We said all living things had irritability or excitability. That means they can respond to a stimulus. Well, neurons are highly irritable. They respond to adequate stimuli by sending action potentials. So if a stimulus is adequate enough to cause a response, we call that threshold. And an action potential may be conducted all the way down the axon. If we reach threshold, this is called an action potential. So when we stimulate a neuron up to threshold, it can fire and send an action potential down the entire length of the axon. You know that your body is electrically neutral, but because we separate charges with plasma membranes, charges gather around that membrane like water behind a dam. We know that inside the cell we have a negative charge, slightly negative charge. Outside the cell we have a slightly positive charge. So separating charges creates potential to do work across those plasma membranes. So there's some regions that are charged because of separation of charges. That's true of a resting membrane or plasma membrane. At rest, the plasma membrane, if you were to measure it with a voltmeter, has a measurement of -70 millivolts. In order to open up channels and cause that muscle to, or that nerve to respond, you would have to change the voltage across to -55, and we can do that through a electrochemical signal, or we can do that by hitting it with a stimulus. Once we reach threshold, then the entire membrane becomes depolarized, and it goes all the way up to 30. So you continue to let sodium ions enter until you get to about +30. Now, because membranes dam up charged particles, it creates potential to do work. Think about a dam that holds back water. If you lift up an opening in the dam and you were to have turbines that were being turned by that water going from high water to low water, then you could make electricity, you could do work. In the body, electric currents that are sent are ions flowing. Instead of water flowing, we have ions flowing. Look at our plasma membrane. You see the double phospholipid layer, and you see how charges are separated. We're negative on the inside and positive on the outside. On the inside of the cell, you have more potassium and less sodium. You also have very big negatively charged protein molecules. On the outside of the cell, you have more sodium and less potassium. Chloride here is showing more chloride outside the cell than inside the cell. And notice that it's negatively charged as well. Plasma membranes contain many ion channels. Some of these are passive leaky channels. That means that they're open all the time, and if the molecule is the right size, molecules can move freely across it from greater to lesser. But other channels are gated. That means that they have a gate that has to be opened with a key. Some gates are going to be opened because a chemical attaches, like a neurotransmitter or a hormone, and so it opens up the channel and ions start flowing. Other channels are voltage gated channels. That means that you have to have a change in the membrane to get them to open. Notice the cell is negative on the inside and positive on the outside, but because it's been stimulated, somewhere it's allowed positives to move in, so this electrical difference causes this channel to open, and now more sodiums rush in. Once the gate is open, the ions flow down the gradient. Since chemicals that are dammed up are charged particles, and because they create a gradient, it is referred to as an electrochemical gradient. So you've separated chemicals, but you've also separated charges. So let's look at resting or resting or polarized membrane. Because the membrane's channels and pumps separate ions, the membrane is said to be polarized. That means that it's slightly negative on the inside as compared to the outside. The difference between the charges on the inside and the charges on the outside is about negative 70 millivolts, but we know that it may range from negative 40 to negative 90, but from this point on, we'll consider our polarized membrane to have a charge of negative 70 millivolts. There's several factors that help us establish this membrane resting potential. One is that there are negative protein on the inside of the cell that cannot move. They're impermeable. They have a negative charge. So there are more of these proteins on the inside of the cell, so that contributes to the negative charge. The membrane is only slightly permeable to sodium. There's more sodium on the outside, less sodium on the inside, so just a little bit is moving from the outside to the inside. It's more permeable to potassium. Potassium is low on the outside and high on the inside, and since it's more permeable, you're going to have more positive potassiums leaking out. So if positive potassiums, more of them are leaking out than positive sodiums coming in, that would make it negative on the inside. And finally, it's freely permeable to negative chloride ions. So the permeabilities of potassium and sodium across the membrane are different. Suppose a cell only has potassium channels. Potassium loss through the leaky channels establishes a negative membrane potential. So we have all the sodiums moving, or all those potassiums moving out, leaving it negative on the inside to about negative 90. So why isn't normal resting membrane potential negative 90? Because you have a few leaky sodium channels, and those few leaky sodium channels are going to allow just a few sodiums to link back in and bring that membrane potential from negative 90 up to negative 70. And finally, we have the sodium-potassium pump, so that it can pump that sodium that leaked in back out and the potassium that leaked out back in. So it helps us to keep that resting membrane potential of negative 70. The electrochemical gradient is mostly due to more potassium leaking out of the cell than sodium leaking into the cell, and the action of the sodium-potassium pump that pumps how many sodiums out? 3 sodiums out and 2 potassiums in. So just think about that. 3 positives out to only 2 positives in. That gives your negative charge as well. So how do we use membrane potentials as signals? Neutrons use changes in membrane potential as communication signals for receiving, integrating, and sending information. The change in potential could be from something that changes the ion concentration or something that changes the permeability of the ion. When these changes occur, they can make the membrane less negative on the inside, so that is depolarization, or more negative on the inside, which is going to be hyperpolarization. So let's look at our membrane potential of negative 70. If we were to open up sodium channels and positive sodiums run into the cell becomes less negative, or it depolarizes. If something happens so that more negatives come into the cell, so it was at negative 70, and let's say that a bunch of chlorides come into the cell, then it becomes hyperpolarized. When we talked about the neuromuscular junction, and we talked about neurotransmitters opening up sodium channels and sodium rushing in and causing depolarization, that's the only direction we went. We were polarized, we depolarized, and we repolarized. But in nerve cells, you have more than acetylcholine. You have some neurotransmitters that inhibit the signal, some of them that open up channels to make it more negative on the inside, further away from threshold. So it's a little bit different when we look at nerves. So if we depolarize, we're getting away from the negative and going towards the positive. If it becomes more negative, it's hyperpolarization. Generally, there are 2 types of signals due to membrane potential changes. One of them is called graded. Remember when we were looking at the neuron, naming the dendrites, the cell bodies, and the axons, we said that what kind of changes happened in the dendrites was graded. These are short-lived. They're only local changes that decrease and magnitudes with distance. They vary in strength. When the ion channel opens, it occurs in the soma and the dendrites. Ion channels are opened by chemicals or by sensory stimulus, not by voltage. So we're talking about potentials, changing the membrane potentials in this area of this neuron. What's going to happen is this one might be stimulated to 10, and this one might be stimulated to 50, and this one might be stimulated to 10. And so 10 and 50 and 10 add up to 70. And so if that's enough to fire, if that's threshold, and that's enough to fire this axon, then the impulse will travel all the way down the axon as an action potential. So when we open up these ion channels, it's because a neurotransmitter opened them, or sometimes a sensory stimulus. It's very short-lived. They're just going to go from the dendrite to that axon helix and then die out. They're very short. So if this is the area where we've depolarized, we're just depolarizing, and we're going to go a little bit down either side, and it's going to die out. It's not going to travel the entire length of the extension or the process.
Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated sodium channels to open, and sodium ions rush into the cell. The cell